All the dignitaries and dear participants, greetings of the day. It is a matter of great pleasure for me that today I got a chance to introduce about an eminent researcher, Dr. R.S. Jayasree. Dr. R.S. Jayasree is currently heading the Division of Biophotonics and Imaging Biomedical Technology Wing, Sree Chitra Tirunal Institute of Medical Sciences and Technology, Trivandrum, Kerala. She has completed her education from Kerala University and Cochin University of Science and Technology. Her areas of research and trust are biophotonics and bionanophotonics for diagnosis of therapy, molecular imaging using optical and MRI techniques, vibrational magnetic resonance and fluorescence spectroscopic methods applied to the biology and medicine. She has published more than 100 research papers spanning in these areas, several book chapters, and has 8 patents to her credit. Major awards and honors received by her are MRSI Medal in 2017, PSN National Award for Excellent Research and Development in Rural Technology 2011, Endeavour Research Fellowship by Australian Government 2009. She is the Fellow of Academy of Chennai and Royal Society of Chemistry, London. It is a proud privilege for us that today Dr. R.S. Jayasree is among us to deliver a talk on the topic surface-enhanced Raman spectroscopy for diagnosis of neurodegenerative diseases and cancer progression. Now I request Dr. R.S. Jayasree to deliver her talk. Dr. Jayasree.
So the topic is surface-enhanced Raman spectroscopy for the diagnosis and the progression, diagnosis of neurodegenerative and progression of cancer. So I thought that being the ROWS, mostly the audience or the listeners would be physicists and mostly with the physics background, and even though I am also a physicist, now I am working in the interface of physics and biology and medicine. So this talk probably will be mostly an application based talk, so where I have chosen being a RIOS in commemoration of the Raman series. So I have chosen the technique of surface enhanced Raman spectroscopy. So I will actually speak on 3 cases here, how this particular technique, very useful technique of surface enhanced Raman spectroscopy could be used for the diagnosis of Alzheimer's disease and for the detection of circulating tumor cells in the case of metastatic cancers, and also how you can use this technique in the case of cancer prognosis when it is under the management. So I will go to the first case where I am talking about the neurodegenerative disease, and being the listeners being the physics background or probably chemistry background, I'll speak a little bit on the neurodegenerative disease. So as you know, many of you know, because these are all known, even though you are not working in the biology area. So these things are known because the neurodegenerative diseases and especially Alzheimer's disease, the incidence is very high nowadays, and which this disease, which was observed at late 60s or early 70s earlier, now we see this incidence in early 50s and or even late 40s. So that way, any common people will be having some idea about the neurodegenerative diseases. So worldwide, an estimated 60 million people suffer from neurodegenerative diseases, and mostly it is, though it is under the umbrella of neurodegenerative diseases, the most common one that we know are Alzheimer's disease, Parkinson's disease, and ALS. And these are devastating diseases, and which takes away a lot of the economy of the country, and also the not only the patient but also the caregivers also find it difficult. So it is a progressive neurodegenerative disease, as you may be knowing, at least some of you may be knowing if you have any acquaintance. So its progression is very slow, though it starts maybe during late 50s or early 60s. Its progression is very slow. So it begins with a mild memory loss and involving parts of the brain that control thought, memory, and language. Here it is written, Alzheimer's symptoms first appear in their mid 60s. So this is a old observation, though I like to put it as mid 60s, because even though we observe in youngsters nowadays. So most common form of dementia is Alzheimer's disease. And according to the WHO, it is the 6th leading cause of death. And it is estimated to be 16 million by 2050. And here you can see the dementia forms for 0.4% of leading cause of death worldwide. This is a 2016 data. So this Alzheimer's disease is characterized by neuronal cell dysfunction. I hope the image, these neurons are visible. If you can look at the neurons, normal neuron, you can very well see the normal neuron and what is wrong with the Alzheimer's neuron. You can just from the picture itself, you can see the differences here, what it is, I'll just explain. And in the axon also, what is the change, and you can see many foreign bodies inside the neurons, and that is one of the main characteristics of Alzheimer's disease. And as the disease progresses, the shrinking of the brain tissue, or in the medical term, it is the atrophy of the brain. You can see the normal brain size, and after Alzheimer's disease, many of the neurodegenerative diseases, you can observe this change as it will shrink the size of the brain. So the size becomes very small. So progressive cognitive motor behavioral impairment happens. Mostly we observe the behavioral impairment, and which ultimately leads to death. So there are different stages of stages and symptoms of Alzheimer's disease. And first one, these are all clinical terms: mild cognitive impairment, where the symptoms are very unnoticeable. So short-term memory loss, probably the patient may feel, oh, I have some memory loss, and the others may not be able to observe or make out the symptoms at this stage. Then mild Alzheimer's disease, the symptom reading problems, poor object recognition, poor direction of sense, etc. So then while it progresses to moderate Alzheimer's disease, it becomes pure judgment, impulsivity, short attention, and later it goes to severe Alzheimer's disease with the symptoms of visual, including visual problems. So the problem with the disease is like, even though you recognize or you diagnose it at the very early stage, this is one of the disease where there is no medicine or treatment so far in the medicine. So why, you will be wondering why the use for early diagnosis. So that is another question, but you can choose the better way of management of the disease, better way of management of the patient. If you know, even at the mild cognitive impairment, if you can diagnose that the patient is going to have Alzheimer's in few years, then we can have a better management strategy. So one or two slides only, I have the introduction. So the main cause of Alzheimer's disease is, you can just see in the normal neurons, if you don't have any foreign body. But in the case of Alzheimer's disease, you see neurofibrillary tangles here. You can see inside the neurofibrillary tangles caused by protein aggregation. And also outside the neurons, you can see amyloid plaques. So these aggregation of these tangles, plaques, and neuronal fibrils forms the main cause of Alzheimer's disease. And why this tangles and neurofibrillary tangles and amyloid plaques forms. There are so many reasons given in the textbooks, which I have not included here because it is a varied reasons. So coming to the biomarkers which we can use for the detection of this disease, these are the major biomarkers that clinically they use to detect that this symptoms are of Alzheimer's disease. The most important ones are the tau proteins, that has different proteins, p tau, t tau, and NFL, and A beta, that is amyloid beta tangles that forms the plaque, and amyloid beta extending from 1 to 42. There are different forms of 1 to 42, and out of which 40 and 42 are believed to be the most important ones. So these are the biomarkers in Alzheimer's disease. So these are the biomarkers that we expect in the normal and Alzheimer's patients. So when you talk about Alzheimer's disease, all of you may be knowing that this is the disease that affects the brain. They have shown the brain atrophy and all. So mostly the biomarkers are present in the cerebrospinal fluid. So for the diagnosis of this disease, the most preferred media is the cerebrospinal fluid, and you all know the difficulty in drawing the cerebrospinal fluid from a patient, and how the patient will be willing, and how the patient will be cooperative, and how the mental condition of the patient, all those things. There are so many challenges in drawing the cerebrospinal fluid. So if you can draw the information from the blood, that will be very good, but the presence of these biomarkers in the blood is very, very, very small. So you cannot just detect these small volume or concentration of amyloid beta or tau protein from the blood if you don't have very, very, very sensitive techniques. So here comes the use of our Raman spectroscopy, and which could be enhanced multiple folds using surface enhanced Raman spectroscopy. So the basis of surface enhanced Raman spectroscopy, I have included 2 slides for the benefit of students who are attending here. So the principle of there are chemical and electromagnetic 2 type of surface enhanced Raman spectroscopy, and most of us are using the technique of surface plasmon resonance based enhancement of Raman spectroscopy, and for that we mostly use the gold or silver nanoparticles, where you see a cloud of surface electron which resonates with the incident rays with the electric field. So these rays increase or enhance the electromagnetic spectrum multiple fold, which could be used for the enhancement of Raman signals, because the Raman signals in general is very weak, and also when the concentration is very, very low, it is very difficult for you to detect these markers. So here I have just shown one slide where you can make out the difference between surface enhanced Raman spectra and those of Raman spectra without surface enhancement, and you can make out the difference between the 2. So what do you want for enhancement of these surface, this Raman spectra? So in many ways you can do that. So the most common way of you doing is using colloidal silver or colloidal gold nanoparticles. But here we have made a substrate of aluminium. We are planning. So this is the schematic. So we have used an aluminium substrate coated with the gold. So gold also, even though gold and silver acts as a very good surface enhancement agent, you can, the advanced science says that if it is a sphere, it will give an enhancement of this much. And if it is a irregular shape, it will give you much more enhancement. So it is better to choose a most irregular structure for the enhancement of Raman signals. So basic theory is given here. You can use the sulfurs, gold or silver in the colloidal form or coated on the substrate. So here you can see the enhancement is very high when you use the, what is that, spheres, but it increases if you use a triangle or a star, where you can see the hot spots and the enhancement formed between at the tip or at the asymmetric edges is very high, and the enhancement is very high when you use most asymmetric forms of nanostructures of these metals. So here we are using gold nanoparticles, which slowly grows into asymmetric gold nano stars. You can see it here, the absorption property and the TEM image and SEM image. And this is the, because we are using these metals nanostructures to be coated on the surface, and over which we have to modify it with antibodies. We have to see the wettability of the surface, which we have measured using the contact angle and optimized it using the coating techniques, modifying the coating techniques. So this is the substrate, the raw aluminum substrate, and which gives a Raman signal, something like that. And we have later on, this is a schematic, we have later on modified this with the gold nano stars, to which we have conjugated the antibodies, and to which the analyte conjugation will be very high due to the antigen antibody affinity. So initially the crystal, the basis of the surface of the the substrate was checked using the normal Raman molecule. It is a crystal violet, and we have got a 10 to the power of 6 fold increase using the nano stars that we have synthesized. And then to that we have chosen two antibodies. Here we have used A beta 42 and 40, and also P tau, that is protein, tau protein, total tau protein, and P tau 181. So these are the antibodies that we have chosen, which are responsible for the cause of Alzheimer's disease. So here you can just see, this is after conjugation of the aptus. Aptus is the modification of the surface. Here you can see, this is how we made this is the gold nano stars. Then through the aptus coated aluminum substrate, which is cross-linked with the glutaraldehyde, and again to which the antibodies are coated. So these are the Raman spectrum at the each stage, to make sure that the stages are complete in the way you want. Then the antibody conjugation was finalized with the HRP assay, where you can see the color change, naked eye color change here. So to make sure the antibody is coated properly. So then we have chosen the two antibodies of interest. Here we have used beta 1 to 42, even though it is beta 1 to 42, amyloid beta 1 to 42, we are using here at 40 and 42, and P tau 181. So this is the Raman spectrum of amyloid beta, and this is of tau protein. So you can just see the enhancement in the Raman signal, and this is the concentration. So this I don't know whether it is visible, the marking here. This is attomolar, this is femtomolar, this is picomolar, and this is nanomolar, this is micromolar. So from micromolar to attomolar, the attomolar level I am not claiming, but up to femtomolar, you could detect the presence of antigen in the presence of this antibody. And this is the amyloid protein, and here it is the tau protein, P tau 181. So we got a enhancement of 10 to the power of 6, so that these are, I would again like to highlight the point that this we are using it only or using the antibody and antigen and not on the or not on the sample from the patient. So this is the preparation of the substrate for the Raman technique. So from that, we identified which are the most important Raman bands that will be useful for the diagnosis of Alzheimer's disease. So then we decided amide 1 band, histidine band, and protein CN band at 1635, 1440, and 1080 are the most useful bands, or the bands which enhances according to our expectation. So then we have used this to check here. Sorry, the slides got shuffled. Here it is very clear. You can see the attomolar is the green one, which is not that attractive, though up to femtomolar you can get the Raman signal very beautifully. That the previous one was, this was for amyloid beta, and this is for p-tau 181, where the attomolar is very weak, whereas in the case of amyloid beta it is slightly better. So in the case of amyloid beta 142, still we can say up to attomolar sensitivity is possible, whereas in the case of p-tau 181 it is not attractive. Then we get the sensitivity only up to picomolar level. So after optimizing all these parameters, and also after optimizing the peaks which we have to look for, we have collected the blood samples from the patients, and we got 3 groups of patients, one of a control patient, that are volunteers, and another group mild cognitive impairment. So that is the earliest stage of Alzheimer's disease, and actually Alzheimer's disease patient, where we have looked for A beta 42, and the first row is here, it is A beta 42, and the second down row is A beta 40. In both cases, we could see a drastic difference between the peaks, which we further analyzed using statistical methods to find out the difference, clear difference between the 3 groups. And this is the p-tau 181. The first one is mild cognitive, sorry, this is the control, which is mild cognitive impairment, and this is Alzheimer's disease. And here it is total tau. The first row is total tau, and the second row is p81. So these are this is the result of 15 patients. We have already done 50 patients. So the analysis we could do only from 15 patients. So you can see the differentiation is very well established in the Alzheimer's disease. For example, you can just see this is the ratio that we have taken. So this is the established ratio, A beta 42 by A beta 40, p-tau 181 by A beta 42, and total tau by A beta 42. These are the clinically established ratios for checking the presence of these antibodies in the CSF. So we have adopted the same for the blood. Here again I am telling we are not testing the CSF. These are all from the blood of the patient. So from the blood, if you can get a sensitivity of the order of picomolar or femtomolar or attomolar, it is very good, because the presence of these markers in the blood is very, very, very less. But even with that very less concentration of biomarkers, you can see that all the ratios shows a very good differentiation between the 3 groups of patients and the control that we have studied. So here it is the significance difference plotted in the bar. It is very clear. And from that we could draw a value for these ratios in the 3 different groups, where you can see 0.732 of the order of 0.732, where it increases to 1.7 in the case of control. So here it is very well established, or it is already known, but using this technique we could derive the information that This is possible with the surface enhanced Raman spectroscopy. So for the value for the Alzheimer's disease, value for the mild cognitive impairment, values for the control are very different. At least these values are very useful to differentiate between the three groups. So this is very good for the clinician's point of view. So that is the inference from the first case. So now I will go to the second case where the Raman spectroscopy, surface enhanced Raman spectroscopy has been utilized for another use. That again, it is a very important field in the cancer management, cancer patient management. So here again, I am taking the blood samples of the patient. So we know that the biopsy for any of the cancer that we suspect, the biopsy is the most important, earliest technique through which the doctors or the clinicians confirm the presence of cancer, whether it is benign or malignant, etc. So that many of the patients may not be willing, and many of the in many of the cases, the diagnosis will be at a very late stage. But drawing blood is not that difficult, and almost 100% of everybody will be willing to give blood. And also for various other reasons, we go to the clinics for testing the blood for various other studies. So drawing blood is not a big thing. But the problem is, again, as in the case of Alzheimer's disease, here also the presence of these cells are very, very less, of the order of 1 to 10 CTC. So, sorry, I will tell you what is CTC. So the circulating tumor cells are found in the blood of a cancer patient. For example, if the cancer is a benign, or if it is a not harmful cancer sitting somewhere without harming anything, so that won't cause any problem for the patient. Probably it will sit there lifelong. So when it is a cancer, or if it is a cancer of malignant in nature, it starts spreading to other organs. Then only we call it as metastatic cancer. So metastatic cancers are, most of the metastatic cancers are known nowadays. The most common one is breast cancer, and another one is colon cancer, etc., etc. So in those cases, the clinicians will be suspecting a metastasis or the spreading of the cancer to other part of the body. In those cases, they can check the blood and find out the presence of the circulating tumor cells. And the presence of the circulating tumor cells in the blood means the cancer has spread to other part of the body. So we can see how this metastatic circulating tumor cells could be detected using our Raman spectroscopy. So we have developed, this is the schematic from the publication where I have published this work. You can just see we are drawing a blood here, and this is the prototype we have made. This is exactly like the 15 ml centrifugal tube on which we have modified a filter. I'll show you in the next slide. And the filter is modified to catch the circulating tumor cells using the materials that are useful to enhance the Raman signal. So this is the actual photo. It has a top chamber, and this is the filter holder, and this is the filter, and the filter is having pores of size 8 micrometer. And why it is so, I'll tell you. And this is the base where we collect the residual blood. So now coming to, I'll show you that again, once again. So on the filter, this filter modification is shown in this slide. On the filter, we have used the graphene gold hybrid. Here you can see it is the graphene and gold hybrid to enhance the Raman signal, and it has been modified with the antibody for circulating tumor cells. So here in this case, the tumor cells, the number of tumor cells being very, very low, we have used two types of antibody. First, we have made a nanotag. So the nanotag contains EBCAM antibody. Here we are using breast cancer cells. We are testing it on breast cancer blood patients. So we have used the antibody specific to the breast cancer. So one is EBCAM2 and another one is ERB2, ERB2. So these two antibodies we have used. One we have conjugated to this nanotag, and another one we have coated directly on the filter, and it makes a sandwich between these two when you collect the blood, so that because of the affinity towards the antibody, the circulating tumor cells will stick on this filter, which is this filter. And because RBCs and the size in the size of the RBC and the circulating tumor cells are very different, the CTCs are bigger than the RBC. RBC will get filtered through the holes, but WBC will stay back because it has of the same size of circulating tumor cells. So then because of the affinity of this thing, only the circulating tumor cell will stick on this, others will filter off. So that is the strategy that we have adopted, and these are the characterization technique. You can just see the graphene sheet and the gold nano seeds here, and that grown into nano asymmetric nano structures. And these are the SERS. So different when you are going for a prototype or a medical device, you need to optimize a lot of parameters. What are the concentration of graphene, what is the concentration of gold, what is the concentration of antibody, what is the concentration of first antibody, second antibody, and various trial and error, various combinations. All those things we need to do finally to arrive at a optimized values of these things. So all these things are given here, and we have chosen the maximum the optimized values, which gives you the maximum enhancement in Raman signal and maximum capture of the CTC on the filter. So I told you we have designed this prototype in the exact way that it resembles the 15 ml centrifugal tube. So here you can see, so that if you want it to be centrifuged maybe for 5,000 RPM for 5 or 10 minutes maximum. So only a centrifuge is required for the separation of this circulating tumor cells, and you can separate this 3 chambers and you can take out the filter, and that the filter could be viewed under the microscope. So these are the parameters again, how much RPM you would need. So here it is the 600, 1200, 1800 different RPMs we have tested, and while testing you should make sure that the RBCs and WBCs are completely filtered off, and also the maximum circulating tumor cells are captured on the filter. So with all those things, we have selected the RPM which is most suitable. Then we have checked the presence of the CTC on the, initially we have checked with the spiked breast cancer cells. Then we could find out up to 5 cells in the blood, we could get signal. For this, the Raman signal that arises is from the graphene. We have chosen the D and G bands of the graphene for the detection of Raman signals. So enhancement of D and G bands enhances with the presence of the circulating tumor cells, because we are holding these materials with the antibody corresponding to these circulating tumor cells. So in addition to the surface enhanced Raman spectroscopy, we could also use the same filter for immunostaining, so that you can use the filter for under the microscope to check the presence of circulating tumor cell. This is the zoomed image. So we could not get 1 cell, but we could get up to 5 cells. But the technique would be used for both surface enhanced Raman spectroscopy and circulating tumor cells. So this is the third case where we have used again a dual hybrid material. Here again we have used gold and graphene. So why we have used gold and graphene? We wanted to have dual imaging come trimodal therapy. So we have used gold and graphene plus porphyrin. Porphyrin is a photosensitizer which could be used for the photodynamic therapy. And because of the plasmon absorption, you can use this gold for photothermal therapy because it absorbs in the visible region. If you are using a laser which matches the absorption of the gold, it converts the energy into heat. That heat could be used for the treatment of cancer. So that is photothermal therapy. When you are using photodynamic therapy or photosensitizers, again, the light or laser corresponding to the particular photosensitizer will produce singlet oxygen, and that will also kill the cancer cells. So these are the two modalities. In addition to that, we have also used the chemical here, chemotherapy, not chemical, the drug, cancer treating drug, doxorubicin. So how we have designed it, it is in a beautiful manner. We have made it in a fashion that this has affinity only towards the cancer cell, with a cancer specific marker attached to this. Here we have used the folic acid so that it will spare the normal cells and it will go only to the cancer cells. Sorry. In addition to that, because we are using the lasers for the photodynamic therapy and photothermal therapy, we can tune or we can modify or we can the sequences could be modified in such a way that this heat released could also be used for the triggered release of the drug. So what happens is when the porphyrin is there, it will give you a lot of fluorescence which may not be useful for the Raman imaging. So what is the intention is you can use this gold graphene for a Raman based surface enhanced Raman based imaging, and the fluorescence from the photosensitizer fluorophobe is used for fluorescence imaging, that is the dual imaging. And the photodynamic therapy, photothermal therapy, and the chemotherapy using doxorubicin, the three-modal therapy. So we should have a sequence how these two is to be. So the cancer cells has a very acidic pH. Once the material is in the cancer cells, as I told you, this goes only to the cancer cells. The laser that we are using corresponding to this, and we have used an 808 laser. It triggers both for photodynamic therapy we used the 535 nanometer laser, and photothermal therapy we used 808 nanometer. So this 808 nanometer laser triggers heat, and at the same time it releases this doxorubicin. While the 635 nanometer laser triggers the photodynamic therapy where the singlet oxygen. So you can choose which one to use first. So first you can finish the fluorescence, then only you need to trigger the material, so that initially the photosensitizers are capped inside the material, so you won't get any fluorescence. So the Raman imaging could be done initially before you trigger, so that once the laser is radiated, the laser is shined, then it will not be possible to get the Raman signal, so that you will get a very high fluorescence. So these are the results of this, and the first one is the Raman signal, this is the fluorescence, and this is the singlet oxygen production, and this is the heat generation. Here you can just see that this is the heat generation using the various stages of the material preparation, and this is the heat generation when if you stop the laser triggering. And here finally is the Raman enhancement of the different pH. Here again you can see the last one at pH 5.5 and pH 7.4. With the material without laser and with the material with the laser, you can see both gives you Raman enhancement. But whereas the material at 7.4 with the material without laser or with the material with laser, both you don't see any Raman enhancement. So this is the last case that I would like to discuss with you, and this has been checked in the cellular media, where these are the different staining to see whether the material has gone inside the cell, etc. And this is the Raman image, where you can see the Raman spectrum here. And ultimately, this has been checked in the animal model, and the photodynamic therapy has been performed in the in vivo model as well. And at a later stage, at 24 hours, starting from 0 hours, the fluorescence images is maximum at 6 hours. Then slowly it goes down because the material will get cleared from the bloodstream. And the animal tissue were collected, different organs were collected, and from the tumor, liver, spleen, kidney, heart, lung, and all other organs. We have checked the Raman signal, and you can see the signal is maximum from the tumor, and the biodistribution also could be checked using this technique, where you can see the Raman signal from the other organs are minimum. That means the material has not gone to other organs, or its biodistribution is maximum in the tumor area, where as per our design, we have intended that by using the cancer targeting material into the conjugating to the material. So with this, I have done the case discussions. So it is not moving the slide. Sorry. So I stop my talk here, and these are my students who contributed to the work which I have presented here, and I am open for any questions and discussions. Thank you.